Next speaker is going to be Irina Alecu from Ottawa, where she is I think, Silverstein Fellow. So thank you very much for coming here. She works on GBA and mitochondria and is in the lab of Michael Schlossmacher and Stephanie Bennett. So welcome and thank you.
Okay. Hi, everyone. Thank you for the opportunity to speak here. So I'm going to be talking to you about GBA1 and mitochondria in the context of Parkinson's disease pathology. So just starting off very simply, GBA1 encodes for GCase, which we all know. GCase is abundantly expressed in the brain, specifically in dopaminergic neurons. We don't quite know why it's so abundant in the brain yet. And it is localized to lysosomes, where it's involved in sphingolipid metabolism, specifically in cleaving glucose from glucosal ceramide to yield ceramide, which you see here, and glucose. So when GBA1 is mutated, this results in a loss of activity and an increase in glycosphingolipids. As we've seen, there's some controversy as to which specific glycosphingolipids are increased, and there are differences in what's increased in men and women. And as we just saw, this drives alpha-synuclein aggregation, and alpha-synuclein aggregation can, in fact, further impair GCase activity. So where does the mitochondria come in here? So let's look first at healthy mitochondria. So mitochondria are very important in cells. They play roles, of course, in ATP synthesis, as well as regulating calcium in cells and in apoptosis. So most of what drives mitochondrial physiology and function is this proton gradient, which is expressed as the mitochondrial transmembrane potential. And this is established via the electron transport chain, which is composed of 5 multi-subunit complexes, with ATP synthase being the 5th one. Complex 1 is the largest and most complex of these. And so what happens is electrons get transferred down the electron transport chain, and in so doing, protons are pumped from the inner membrane space into the, from the matrix into the inner membrane space. And this energy, this free energy stored as the proton gradient, is then used to drive ATP synthesis. So neurons have particularly high energy demands compared to other cells because they need energy for release and recycling of synaptic vesicles, re-equilibration of ion flux, transport of molecules over the large length of the axons, as well as for synaptic regulation via calcium clearance. In healthy cells, unhealthy or dysfunctional mitochondria get degraded via mitophagy. So when the proton gradient or the mitochondrial membrane potential collapses, this recruits PINK1, which then phosphorylates ubiquitin and parkin. And these then recruit the autophagosome, which then transports the enveloped mitochondria to the lysosome for degradation. So the first clues that mitochondrial dysfunction is implicated in PD pathology came in the early 1980s, when it was found that this toxin, MPTP, which was a contaminant of heroin, was giving heroin users Parkinson-like symptoms. And in fact, in autopsies, they found that the neurons in the substantia nigra were degenerated in these patients, in the heroin users. Furthermore, subsequent genomic studies revealed that mutations in mitochondrial genes like PINK1 and parkin, which we just talked about, increase the risk of Parkinson's disease, further validating the link between mitochondria dysfunction and Parkinson's disease pathology. But what is the link specifically between GBA1 PD and mitochondria? So this field only started being worked on about 11 years ago. So the first papers linking GBA1 and mitochondria came out in 2013. So it's a fairly new field. And what we know so far about the link between GBA1 and mitochondria is that in impaired GCase models, so heterozygous and homozygous knockouts, or knock-in mutant models, we know that complex 1 activity is impaired, which you can see here. So it then follows that complex 1 activity is impaired. There's also loss of mitochondrial membrane potential, so this proton gradient can't be maintained. And this again has been shown in a variety of mouse models. And again, it follows that if the proton gradient is not maintained, ATP synthesis is decreased, which you can see here. And furthermore, Osalame et al. Showed that in fact there is reversal of ATP synthase activity. So instead of the ATP synthase pumping the protons from the inner membrane space into the matrix, therefore producing ATP, it's actually pumping protons the other way. So as we see, the mitochondria is dysfunctional when when GCase is dysfunctional, and dysfunctional mitochondria produce reactive oxygen species. And this has, in fact, been shown when GCase is impaired, either by inhibiting it chemically or in GBA1 PD patients as well as mouse models. So when the electron transport chain is not functioning properly, electrons leak out of the membrane and produce the reactive oxygen species. Reactive oxygen species have then been found to produce oxidized dopamine. This was first shown in DJ1 knockout neurons. So these neurons have increased oxidant stress because DJ1 is knocked out. And what was found in this neuron is that they accumulate oxidized dopamine, and this was also in fact shown in GBA1 PD neurons, which you can see here. And this oxidized dopamine, in fact, was found to inhibit GCase activity further. So we know mitochondria are dysfunctional. They're producing reactive oxygen species, so they should be degraded via mitophagy in principle. However, it also appears that mitophagy is defective when GCase is defective. So what we see is that there's no autophagosome formation or recruitment in GBA knockout mouse neurons. And furthermore, there's no recruitment of Parkin. So because the membrane potential is dissipated in the GBA knockout mouse neurons, we should be getting recruitment of Pink and Parkin and of the autophagosome, but this is not in fact happening. And we see a decreased expression of Parkin in mutant mouse neurons as well. So autophagosome recruitment is impaired, and so it follows then that degradation via the lysosome is impaired. In fact, we don't see colocalization of mitochondria and lysosomes in GBA mutant mouse neurons as well as in GBA1 PD patient neurons. Furthermore, their mitochondrial size and morphology is altered in the context of GBA1 mutations. So you can see the mitochondrial cristae are disorganized instead of lamellar. There are higher numbers of fragmented mitochondria and significantly smaller mitochondria in the context of GBA1 mutations. Furthermore, as I said, mitochondria are involved in calcium buffering in the cell, but this also appears to be impaired in the context of a GBA decrease or GCase decrease in activity. So what you can see here is when neurons and astrocytes are given a metabolic stressor, glutamate, instead of a transient calcium increase that then goes back to homeostatic levels, in both heterozygous and homozygous knockouts, what we can see is that calcium levels in the cell do not go back to homeostatic levels. So typically when there's an increase in calcium in the cell, mitochondria via the mitochondrial calcium uniporter then takes up this extra calcium. However, this is not happening when GBA1 is knocked out. And in fact, the expression level of this calcium uniporter is significantly decreased in heterozygous and homozygous knockouts. So it's also been fairly recently found that there are contact sites between mitochondria and lysosome that are important for the network and dynamics of both of these organelles. And what's been very recently shown is that this tethering between lysosomes and mitochondria is disrupted in GBA mutations. So what happens is the length of time that mitochondria and lysosomes are tethered is significantly increased. And this was also shown upon chemically blocking GCase activity. So GCase activity has a direct impact on the length of time of tethering between the lysosomes and mitochondria. The protein responsible for this is TBC1D15, and this is a Rab7 GTPase. So it hydrolyzes GTP to untether these two organelles. However, we see that this is significantly decreased. The expression level of this protein is significantly decreased in the context of GCase deficiency. So everything I've talked about so far is associative or correlative, right? We see it happening at the same time, dysfunction in mitochondria and loss of GCase activity. But is there a direct interaction between GCase and mitochondria? So very recently, Baden et al. Reported that GCase localized to mitochondria, and they're in fact imported by mitochondrial import machinery. So they found interactions with all steps of the mitochondrial import machinery. So with the TOM complex, which transfers protein across the outer mitochondrial membrane, as well as with the TIM23 complex, which transports them across the inner mitochondrial membrane. And they also interact with the mitochondrial quality control machinery, so the chaperones like HSP60 and LONP1, which has both degradation and chaperone activities. And what you can see here is that specifically in the L44P GCase mutant, so the more severe mutant, there is a significantly increased interaction between GCase and these mitochondrial quality control proteins. Furthermore, it was also reported in the same paper that GCase in fact directly interacts with TIMDC1, so this is a complex 1 assembly factor, and it also directly interacts with subunits on complex 1 like NDUFA10. And these interactions were significantly decreased in the GCase mutants, so it appears to be necessary for complex 1 integrity and function that GCase interacts with both complex 1 and the complex 1 assembly factor. So now I want to come back to lipids because GCase is involved in lipid metabolism. So I think we're all very focused on looking directly at the part of metabolism that GCase is involved in. I want to kind of expand our view a bit to other lipids because there can be side effects of GCase lower activity. So I want to ask if there's an effect of lipid dysregulation specifically in mitochondria, what's happening to the lipids in mitochondria, and are other lipids besides glycosphingolipids affected? So I want to bring up here the deoxysphingolipids. So this is a subclass of sphingolipids that is neurotoxic, in fact. And so SPT, as you heard Peter talking about, serine palmitoltransferase, that is involved in de novo synthesis of sphingolipids, typically takes serine. However, it can also take alanine in the condensation reaction. So if SPT de novo synthesis is triggered, it could also be triggering increased production of these deoxysphingolipids. What's different about these is that they're lacking the OH group on the first carbon, so they can't be converted to complex sphingolipids like glycosphingolipids. So why am I bringing them up in the context of GBA1 or GCase in that case? Well, we found, in fact, that deoxysphingosine abundance, so this is the most downstream lipid in that pathway I just showed you, is significantly increased, specifically in GBA1 PD. What does this have to do with mitochondria? Well, in fact, it's been reported that these deoxysphingolipids localize specifically to mitochondria, and they result in mitochondrial degradation, loss of the cristae morphology, decreased ATP production, and decreased respiration. So all of the same phenotype we see in mitochondria in the context of GBA-PD. So these could also be involved in the mitochondrial pathway pathomechanism. So just to kind of summarize everything, there are a lot of impairments which have been linked impairments between GBA1 and mitochondria. So there's energy production disruption, there's oxidative stress that leads to damage of mitochondrial DNA, which further damages mitochondria, accumulation of oxidized dopamine, calcium homeostasis dysregulation, which can make neurons more vulnerable to increased energy demands, disrupted mitochondrial lysosome tethering, quality control impairment so that there's an accumulation of defective mitochondria. And more directly, it seems that there is disrupted complex 1 integrity, and we're still working on this, on quantifying lipids directly in mitochondria, but there could be lipid dysregulation directly in mitochondria that is affecting their function. We don't quite know yet, are these primary generators of neurodegeneration, or are these secondary factors? So we have to work on kind of the time sequence that everything is happening in. And now I want to look a little bit at whether these mitochondrial impairments can be rescued and how we've tried rescuing them. So because complex 1 is impaired, as I said, NAD+ levels are lower. And so Schondorf et al. Increased NAD levels by giving an NAD+ precursor, nicotinamide riboside, and they found that this rescued the cristae morphology, which you can see here. It also decreased the amount of reactive oxygen species and increased the expression of chaperone proteins like HSP60, which could be refolding GCase. And in fact, treating with nicotinamide riboside was found to rescue neuronal loss and motor deficits in GBA1 flies. Rescuing of the mitochondrial lysosome tethering phenotype was done by increasing expression of this TBC1D15. This also rescued mitochondrial density in the axons, which you can see here, as well as ATP production. And another strategy, which is very popular now, is to augment GCase activity. So one way to do this is with small molecules. In this example, S181 was used. And what was found was that by increasing GCase activity, which you can see here, the levels of oxidized dopamine were decreased. And so kind of this vicious cycle of further inhibiting GCase activity was broken. Alpha-synuclein levels were decreased. And also the tethering, the disrupted tethering between mitochondria and lysosome was corrected by augmenting GCase activity. Lastly, we can also augment GCase activity, of course, by giving recombinant protein. So this is in peripheral blood mononucleocyte cells from Gaucher disease patients. And what was found here by Ivanova is that autophagy was rescued, so autophagosome formation. However, lysosome activity and degradation was not rescued, nor was ATP production. So this was with treatment in vitro. Interestingly, giving this treatment directly to patients had a lot more contrary effects. So whereas it worked in some patients, but not in other patients. So just to kind of summarize, there's a variety of kind of therapies that are being tested out, like enzyme replacement therapy and chaperone and GCase activators. Again, just to kind of reiterate what was said this morning, we don't know if there are interactions between kind of defective GCase and normal GCase. We don't know what this would be doing in the cells. And I think we also need to take into consideration in the therapies, specifically also with substrate reduction therapy, what other lipid pathways are being affected by doing this. So just to end off, what I believe are current gaps in the field of mitochondria and GBA1, we don't know the chain of events that leads to or arise from mitochondrial dysfunction. So what order does this happen in? We should be doing longitudinal assessments of disease progression and mitochondrial function and dysfunction so we can link disease progression with what's happening in mitochondria. We need to understand the differences in mitochondrial pathways that lead to neurodegeneration with different GBA1 PD mutations. Again, this was brought up earlier this morning. I think this is really important to link the mutation to exactly what's happening. And also, again, determining whether the disease mechanisms that drive pathology are time invariant or not. So at what point does mitochondrial dysfunction come into play? Is it resulting in neurodegeneration or is it resulting from neurodegeneration? So I'd like to thank Dr. Bennett and the Bennett Lab, as well as specifically the Silverstein Foundation, the Michael J. Fox Foundation, and the Parkinson Research Consortium. So thank you for your attention.
Thank you, Irina. We have time for a few questions. Yes?
I note that you mentioned using NR as an intervention, but I'm wondering if you've explored interventions for the TBC1D, enhanced expression.
So, yes, so this. Yeah, so increased expression, so this was done by Kim and Kranich et al. So increased expression, what they showed was that it did rescue mitochondrial density, so the number of mitochondria in axons. It increased ATP production also. And it, of course, decreased the length of time between the contacts, but I think that's all they looked at in terms of the mitochondrial function.
No, I was just asking from a patient perspective, in person. So I can give my patients NR and do, but I'm just saying, looking at TBC1D, what interventions?
Oh, yeah, you would have to increase. This is very novel. So in terms of patients, this hasn't been used in patients. I wanted to show you at the molecular level kind of what's happening and to take into account kind of other aspects of mitochondria dysfunction, not just ATP production.
Heidi.
Thanks, Irina. It was really amazing, and thank you for talking about mitochondria. so one of the things that I've been thinking a lot about in the last little while is the contact sites between mitos and lysosomes. The primitive, primordial, initial function of that is for iron flux, right? To develop iron sulfur cluster proteins, or in the mitochondria that are part of complex 1 and all of the and elsewhere, of course. So if you have a GBA mutation, your lysosomes are going to be messed up. The lipids are screwed up. Has anyone looked at specifically at iron trafficking in these situations? Because when you turn again to the dopaminergic neurons, in the biosynthesis of dopamine, it's heavily iron mediated, which is why you have neuromelanin, which Dimitri has also talked about for a long time. People don't talk as much about iron as I think they should, especially thinking about mitolysosome contacts.
Yeah.
So can you comment on that?
So as far as I'm aware, I don't think it's been looked at what's happening at these contact sites in terms of the iron flux when GCase is impaired. I think this is a pretty recent finding, so we do need to look more into it. I think we also need to look more into what else is being fluxed or being transferred at these sites, like the lipids.
And protons.
Yes,
But-
yes, and proteins.
And
I'm a
also just
lipid person,
ferroptosis.
so
I mean, in terms of if you do have mishandling of the iron, right, you can start to accumulate it
Yeah.
in the cytosol. It depends on what's happening with ferritin. But in the dopaminergic neurons, there's been a number of papers lately focusing on ferroptosis as a type of death in those conditions. Thanks.
Okay, up there.
So the work of Baden et al. that you mentioned would sort of force us to think kind of differently about GCase, that is sort of as well an intra-mitochondrial protein. So since you mentioned it, I was wondering, have you sort of isolated mitochondria, say from mouse brain or so, and tried to see that you try to detect GCase in those samples?
So we have actually isolated mitochondria from patient brains. We're not going to be measuring GCase activity because you can't in the post-mortem brains. We are going to be measuring lipid levels in specifically isolated mitochondria to see what's going on with the glycosphingolipids directly in mitochondria.
Yeah. But, I mean, you could do, let's say, see whether GCase is there by Western blot.
Oh, yes. Yeah, definitely.
And
We will be doing that, yes.
oh, you will be doing that.
Yes,
So you have
we
not done it yet.
But we have isolated mitochondria, but we are working on looking at directly at isolated mitochondria, at lipids and GCase, yeah.
Okay. Thank you.
Maybe I can just follow up on that. Do you think that the GCase intramitochondrially would be active given the pH? And what would its substrate? I mean, how would it? Have you been able to measure levels of substrates and?
I think it may have a different function in mitochondria in terms of the lipid metabolism as well. So given the pH, I would expect that it's not active, but it could have a different function in mitochondria. Like, for example, in assembling complex 1. So it might not even be a lipid function, it could be a protein-related function.
Thank you very
Thank
much
you.
for that talk.